Hi, this is Shamin here. In this video, I'm going to teach you biology, that is A-level biology, or you can say Cambridge AS level, that is chapter 1, cell structure. Alright, so let's get started. So in this video, I'm going to give you some question answer only. So number one question, what is a cell? What is cell? Cell is a unit of life. It consists of mass of living matter called protoplasm. So what is protoplasm? Protoplasm is a complex jelly-like substance, that is jelly-like substance which endless chemical activities are carried out that ensure the life of a cell. What is a cell biology? So cell biology, that is the study of cells. Study of what? Study of cells has given rise to an important branch of biology that is known as cell biology. So here is a one broad question, that is name the structures that animal and plant cells have in common. Those found in only plant cell and those found only in animal cells. So I'm going to show you, this is a diagram. So if you see the diagram, this is a animal cell. So what you will see here? Animal cell. So if you see the animal cell, here is a Golgi body, cytoplasm, chromatin, mitochondria here, cell surface membrane here, nucleus, here is a centriole. All right, so this is an animal cell. So in animals and plants, which one is a common here? So animal and plant cells each that is surrounded by cell surface membrane, and it also sometimes referred to plasma membrane. Many of the cell contains colorless and transparent, so they need to be seen. So each cell has a nucleus, that is very important, which is relatively large structure that strains instantly, and there is a very conspicuous, the deeply straining material. The nucleus is called chromatin and is a mass of loosely coiled thread. Another one we can see, that is mitochondria. So mitochondria are only just visible but films of living cells taken with the light micro. If you see the under the light microscope, so you can see properly, shown that can move and change shape and divide. And use of special strains containing silver enamel, that is Golgi apparatus, to be detected for the first time, that is 1898 by Camillo Golgi. So Golgi apparatus is a part of a complex internal sorting or distribution system within the cell. So materials, that is the material between the nucleus and cell surface membrane, is known as cytoplasm. So cytoplasm is an aqueous material varying from fluid to a jelly-like consistency. So what is the difference between animal and plant cell? That is difference. Plant cells are often larger than animal cells, although cell size, that is the varies enormously, and under light microscope the centriole appears as a small structure close to the nucleus, and light microscope, that is individual plant cells are more easily seen than animal cells because they are usually larger and unlike animal cells, and that is they are surrounded by cell wall outside the cell surface membrane. So this is a plant cell. Plant cells are that is the link to neighboring cells by means, that is the fine strands of cytoplasm called, what's that? That is plasmodesmata, which pass through pore-like structures in their wall. Another thing we can see, that is vacuole. So vacuole are not confined to any plant cells, yes, and animal cells may have small vacuole. So phagocytic vacuoles, although these are not usually permanent structure. So chloroplasts are found in the green plants. That is the another difference. And plant cell, you can see chloroplast, that is the green color. So this is a structure plant cell, Golgi apparatus, here is a cell wall, chloroplast. So you draw some diagram and practice. So here is the difference, another difference I'm making for you, that is the plant cell and that is the animal cell. Plant cell have cellulose but do not have cellulose, and here is a plant cell chloroplast but it's do not have, and always have vacuole, and it's sometimes have vacuole and have a few different shapes, have many different shapes. Animal cells. What does the cell nucleus do? What does the cell nucleus do? So nucleus controls what cell does and how it develops. Another question number 5. What are chloroplasts and what do they do? What are chloroplasts? What are chloroplasts and what do they do? So chloroplasts are tiny disc full of green substance. They trap the light energy that plants need for making food by photosynthesis. Explain what a specialized cell is. So some cells become specialized muscle cells, for example. So muscle cells, that is the tissue which can tighten up and relax, and nerve cells are specialized, that is the form tissue which can carry message around the body. Until of measurement, sorry, here is another, that is units of measurement. So 1000, that is 0.0019 micromillimeter, and 1 millions, that is 0.000, how many zero here, 5, that is micrometer. 1000 million, that is so many zero here, nanometer. So calculating the magnification of photograph or image. Magnification, so what is the meaning of word magnification? So magnification is the number of times larger an image than the real size of the object. So magnification, that is the formula, you must remember, that is the observed size of the image divided by actual size, that is m equal to I by A. I means here is a observed size of the image and A equal to actual size. Suppose we want to know the magnification of the plant cell, alright. So plant cell leveled in the figure. So if we know its actual length, we can calculate its magnification using the formula. So we know the formula. What's the formula? Formula is here. That is formula is m equal to I divided by A. I here observe size of the image and actual size. First you observe the image, then its actual size. So here is a step. Step number 1, what you will do? Measure the length in millimeter of the cells. Cells in the photograph using a ruler. So it's about 60 millimeter, and step number 2, convert millimeter to micrometer. So 1 millimeter equal to, you must remember this one formula, 1 millimeter equal to 1000 micrometer. 60 millimeter equal to, that is 60,000 micrometer. Step number 3, what you will do? Step number 3, that is use the equation to calculate the magnification. That is magnification, that is the formula I already given, that is m equal to image size divided by actual size, that is m equal to I by A. So I here is a 60,000 micrometer divided by, that is actual size 80 micrometer. So you just calculate, then you get the answer. Next one, so this is an image, guys. Here is a one image, here is another image. So photograph of the same type of plant cell seen a light microscope and B an electron microscope. So both shown a magnification of about, that is, here is a calculating magnification from a scale bar. So this is a lymphocyte, that is 6 micrometer, a lymphocyte. So first we can calculate the magnification of the lymphocyte by simply using the scale bar. All you need to do is measure the length of scale bar and then substitute this, then the length it represents into the equation. So here is a convert, here is a step number 1, measure. What's the step number 1? Step number 1, measure the scale bar, that is 36 millimeter. Convert millimeter to micrometer, that is the same, guys. Here is another, so then you get the answer. What is resolution? That is very important question. What is resolution? So resolution is the ability to distinguish between two objects very close together. The higher the resolution of an image, the greater the detail that can be seen. So if I make it bigger, then you can see properly, right? So that is the, how much clear is this, the picture, you can see properly. So if you use nowadays, people many years ago not much my electron microscope, but nowadays very advanced level microscope. So what is magnification? That is already I given. Magnification is the number of times greater than that an image is than the actual object. That is magnification. Here is another question. Explain why ribosomes are not visible using a light microscope. What's the reason? Because ribosomes are far too small to have any effect on the light waves. So the general rule is that the limit of resolution, what's that? What's the reason? That is the limit of resolution about 1/2 the wavelength of the radiation used to view the specimen. In other words, if an object is any smaller than half the wavelength of the radiation used to view it, it cannot be seen separately from nearby objects. This means that the best resolution that can be obtained using a microscope that uses visible light in that is 200 nanometers, since the shortest wavelength of visible light, that is 400 nanometers. So in practice this corresponds, the maximum useful magnification is about 1500 times. So ribosomes are approximately 25 nanometer, that is in diameter, that can thereby near that is therefore never be seen using light. All right, so that is the answer, guys. It's details, you can make it short if you want. So here is some question. Which one of the following cell structures can be seen with a light microscope? Which one? The use of electrons as a source of radiation in the electron microscopes, always high resolution to be achieved, because electrons, what's the answer? Write down the answer and check it how much marks you get. Which one of the following structures is found in animal cells but not in plant cells? Which one? So here is another question number 4. Copy and complete the following table which compares light microscopes with electron microscopes. Some boxes have been filled in for you. So is some box filled for you for to understand how you can fill it. That is the source of radiation. Light microscope, what's the source of radiation? Light. Electron microscope, what's the source of radiation? Electron, right? Wavelength of radiation used here, maximum resolution, lens. Light microscope we use here, light microscope, what do we use? Lens, glass. But how about electron microscope? Specimen, non-living or dead, that is the specimen we use here. Stain, that is color dyes. Image, all right, so I will give you the answer, check it if you understand or not. Here is a number 1. Which one of the following cell structures can be seen with, that is mitochondrion? The use of electron as a source of radiation in the electron microscope, that is allows high resolution to be achieved because electron have a very short wavelength. Which one of the following structures is found in animal cells but not in plant? That is cell surface membrane. And here is the answer, guys. That is source of radiation, that is light microscope, electron microscope. All right, electromagnets. Here is a more question, that is what's the difference between question number 6, advise and answering question 6 if you are asked to distinguish between two things. So here's a two things, that is magnification and resolution. I already given the answer, you know that. Light microscope, that is also I already given. Nucleus and nucleolus, chromatin and chromosome. What's the difference between here's the two things, prokaryote and eukaryote. Smooth RNA, you go to your textbooks and read it and find out and practice. Here is a more question here. Manufactures lysosomes, manufactures ribosomes, site of protein synthesis. So I will give you this answer, guys. Here is my answer. Magnification, that are already given. Light microscope use light as a source of radiation. Electron microscope use electrons as a source of radiation. Nucleus is the largest cell organelle, and nucleolus, that is gene control the activities of the cell and inheritance. Those that nucleus controls the cell's activities. Chromatin and chromosomes both contain DNA and both found in nucleus. Chromatin is the loosely coiled form of chromosomes. So chromatin is the form of, that is exist between cell and nuclear division. Another difference, that is what is the difference between nuclear envelope and membrane? So envelope, that is the two membranes situation closely together and surround the surround, it will be D here, sorry guys, the nucleus, and envelope is perforated with nuclear pores, and in animals and plants each cell is surrounded by a very thin cell surface membrane. What is the difference between rough ER and smooth ER? So rough, that is called because it's covered with very tiny organelles called ribosome. Very important, ribosome. And smooth ER, that is called because it's lack ribosomes, has a completely different function. It makes lipid and steroids, estrogen and testosterone. Organisms that lack nuclei are called prokaryotes, and organelles whose cells possess nuclei are called eukaryotes. Nucleolus, ribosome and centriole. That is the 7 number answer. 8 number, that is manufactures lysosome, that is Golgi apparatus. Manufactures ribosome, that is nucleus. Site of protein synthesis, that is ribosome. Can bud of vesicles which form of Golgi body, that is rough ER. Can transport newly synthesized protein round the cell, that is rough ER. That is the answer. Number F, manufacture ATP in animal and plant cell, that is mitochondrion. Controls the activity of the cell which contains the DNA, that is nucleus. Carries out photosynthesis, chloroplast. Can act as a starting point for the growth of spindle microtubules during cell division, that is centriole. Contains chromatin, that is nucleus. that is partially permeable barrier, only about 7 nanometer thick, that is membrane, and organelle about 25 nanometer in diameter, that is ribosome. So here is a more MCQ question that is already given. Is there any new question? That's all for today. Thank you for watching, and don't forget to follow me at Twitter, Facebook page, Instagram, that is Educator Sharman, or you can follow me my website, that is www.educatorsharman.com. That's all. Thank you very much, and good luck your biology test. Thank you so much.